Moving on to our next presenter, Ruby Sharif, presenting Keratocomus, Blurry Vision, Hormones Take the Blame.
A slight change in vision is nothing to worry about, right? We write it off. Too many hours in front of my computer, or I'll check on it later. That blurry vision could be a serious eye disease called keratoconus. 200,000 Americans suffer with this condition every year, and many don't even know that they have it until it's too late. Keratoconus causes progressive thinning and bulging of the cornea, giving it a conical shape. This change in corneal structure leads to loss of transparency and sight. So if you had keratoconus, this is how you would see me today. Currently, there's no diagnostic lab test for this disease because we don't know what causes keratoconus, and we have no animal model to study it. Therefore, it's often misdiagnosed as nearsightedness or myopia. It begins during puberty and stabilizes when patients hit their 30s and 40s, endorsing our hypothesis that changes in sex hormones or proteins related to sex hormones may contribute to this disease. Keratoconus treatment begins with contact lenses, but ends in corneal transplant. And the sad reality is that these options create medical complications, financial burdens of $25,000 on average, and an emotional burden of slowly going blind. This disease robs the sight of many teens, and if left untreated, a corneal transplant is unavoidable. Keratoconus patients desperately need a diagnostic test for early screening and treatment. And emerging evidence from our lab has identified a hormonally regulated molecule named prolactin-induced protein, or PIP, which could be the biomarker these patients need. With this diagnostic marker, we may be able to test these patients before it's too late. In order to research and validate PIP as a potential biomarker for keratoconus, I analyzed tears, serum, and saliva from over 200 keratoconus patients. And what I have discovered is that these patients have low protein levels of PIP, independent of gender. They also have increased levels of DHEA, a precursor to male hormones, and reduced levels of two female hormones, estrone and estriol. This suggests that PIP is involved in corneal homeostasis and that male hormones worsen keratoconus while female hormones are protective. These results are so exciting because our research is the first to proclaim PIP as a keratoconus biomarker and also study sex hormones' role in keratoconus, which may explain the onset and rapid progression of this disease around puberty. These patients see the world through keratoconus, and through our research, we can help them see the world picture perfect. My hope and my goal is that for millions of people, this image can become clear. Thank you.